Our first speaker this morning will be Stephen Hatfill.
Thanks, Art. Good morning, ladies and gentlemen. Can you hear me in the back there? Yes. Okay. What I want to do this morning is take you on a little medical mystery. And it spans several hundred years. It's a mystery worthy of the attention of Sherlock Holmes. And it's a mystery that's still underway. It spans 250 years. It begins with itching sheep in Iceland, human cannibals in the Pacific. It migrates to corneal transplants and growth-stunted children in France, starving deer in the forests of North America, drooling cows in the fields, and young adults dying of a very strange, mysterious neurological disease. The story begins in 1755 when a very strange new disease appeared in the herds of sheep in Iceland. It began decimating the farmers' flocks. The animals began to tremble and to stumble while they're walking. And this was all accompanied by uncontrollable itching of their skin, to the point where they would scrape all the wool off, find a hard object or the bottom of a fence, and just completely scrape the skin raw. It had never been seen before. The ranchers began to call the disease scrapie. While it appeared to be infectious to other sheep, humans seemed to be unaffected. It didn't seem to affect the meat. No clue to its cause or appearance was ever proposed, and the condition languished as a veterinarian medical curiosity for years and years and years. Over the next century, medical science had advanced enough we could look at tissues under the microscope and begin to describe their morphological appearance. And in 1898, French scientists were able to show that the sheep with scrapie had thousands and thousands of tiny little microscopic holes in their basement material, in their tissue, on their brains on autopsy. In 1926, French scientists determined that the brain material from these affected sheep was infectious to other sheep, and that the incubation period for the disease was very prolonged, average of 18 months from the time of exposure to the time of symptoms. Again, quite a mystery, but it sort of languished in the back of the journals of veterinarian medicine. In 1913, and you must remember the Germans at the time were ahead of top in pathology and chemistry. And in 1913, a young neurologist named Dr. Hans Gerhard Creutzfeldt examined a 23-year-old young girl, and she'd suffered from continuous tremors, loss of coordination, hallucinations, leptogenic fits. She was admitted to the hospital in Breslau, and she eventually died from continuous seizures. On autopsy, it was found that this young woman's brain was riddled with thousands of little microscopic holes. Now today, when we see something like this in a tissue slide, we tend to associate it with a process called programmed cell death. Every cell in your body contains a genetic program to tell the cell to kill itself if certain conditions are met. We have cancer cells every 3 or 4 minutes in our body. This keeps this under control. The cell, something's not right. It uses metabolic energy to make enzymes that chews up its own DNA, and uses ATP and metabolic energy to bleb off. And it's a very common process in nature. It's how the tadpole loses its tail. Or during fetal development in humans, there's a little phase where you have a little short tail, and webbed fingers and toes, and the webbing just, at the right time, just suddenly disappears. And it's no inflammation, it's just the cells in that area kill themselves. So this is what something is telling this brain tissue and activating it and causing it to commit cellular suicide. Apoptosis, Greek for a falling leaf. Well, more cases of this were reported over the next 7 years. World War I intervened, and that delayed everything. Obviously, German scientists, they were sent to the front. And the findings weren't published until 1920. By then, Dr. Alfons Jacob at the University of Hamburg had identified 4 more cases. And in 1921, the papers came out, and the disorder was formally named Creutzfeldt-Jacobs disease, or CJD, is what we'll call it in this talk from now on. And there it languished in the backwaters of neurological science till around 1957. Workers in Papua New Guinea began to report the occurrence of a mysterious disease in the highlands among the Foray tribe. The local natives called it kuru, which means trembling and fear in their language. And it was very curious. Victims of all ages showed progressive uncoordinated movements, trembling. They were unable to stand. It seemed to be chiefly affecting the cerebellum, which is the tiny little brain underneath our big brain. And they couldn't stand unsupported, and eventually losing brainstem functions such as the ability to swallow. In 1959, a U.S. pediatrician named Carlton Gajicek and a team of anthropologists arrived in this tribal area in the highlands to investigate. They found, first of all, that the disease appeared to affect women and children only, both male and female children. But the adult males were unaffected. And Gajicek went in and he made friends with the tribe. He lived there. He started clinics. He did all sorts of projects. And he was able to do some autopsies and to get brain material back to the National Institutes of Health, where he was originally from. And the tissue slides made from this material showed these same sponge-like changes with these tiny thousands of little holes, although not as pronounced as Creutzfeldt-Jacobs disease, and also this abnormal amyloid appearing plaque-like deposits in the brain tissue. This is more similar to Alzheimer's and the amyloid protein there. Anyway, in a very, very detailed bit of anthropological work, investigators were able to show that kuru was infectious to humans. Females and children were conducting ritual cannibalism. It used to be between tribes, and that went away. But when relatives would die, the corpses would be eaten, and the brain material. And the adult males didn't participate in this practice. And the females knew that this was what was causing this. And the males wouldn't listen, as always. So they got funding, and they started a primate research center, non-human primate, chimpanzees, outside of Bethesda in Washington, D.C. And the monkeys were intracerebrally injected, anesthetized, and intracerebrally injected with Foray brain material that had remained frozen in liquid nitrogen and brought all the way back to the United States. And this was a long-term project. And I was fortunate enough to sit down with Dr. Gibbs, his associate, about 20 years ago. And these were very dedicated scientists. Dr. Gibbs passed away a little while ago. But of course, you get very attached to these animals, and it takes a toll on the researcher. But 2 years later, after intracerebral injection, they started to develop the classical signs of human kuru, the shivering tremors and the progression and inability to move and inability to swallow. A German pathologist came over to join the team, and on the postmortems he discovered rampant lesions consistent with kuru, establishing that, it was transmissible. He had obviously trained in Germany, and he realized that these chimp brains were consistent with the Creutzfeldt-Jacobs disease, CJD. The chimp brain material was inoculated into other normal chimpanzees, and in less than a year, these animals also developed kuru lesions. So the evidence is getting fairly consistent. And he was also the first to make the link between sheep scrapie and Gajusek put it all together that kuru, CJD, and scrapie were all variants of the same brain pathology. And the causative agent remained unknown. Throughout the 70s, a lot of speculation on that these were lentiviruses, slow and apparent virus infections. There are still some researchers pursuing this line. You must never close your mind in science. And a lot of people remain quite open to this. In the 70s, it emerged again as an iatrogenic infection. There were 2 corneal transplant cases that went on to develop CJD in France. And then a series of 10 small stature out of the 3rd centile French children were injected with an early growth hormone preparation. Now, at the time, we didn't have widespread molecular biology, and it was from pooled pituitaries, anterior pituitaries from cadavers, and the growth hormone was extracted. You can't use cow, like insulin, you can't use cow material. It has to be human-specific growth hormone. That's all that'll work in the human. So these children were injected in an attempt to increase their stature, and they uniformly all came down with Creutzfeldt-Jacobs disease. By the end of the 70s, an unusual disease started to appear in the U.S. deer and elk populations called chronic wasting disease. And this was eventually identified as a CJD type of disorder in elk. It's been gradually spreading from free-ranging and captive animal herds in 25 states in the U.S. and Canada. And again, it's typified by abnormalities in gait, staggering, inability to swallow, chronic weight loss, eventually leading to death from starvation. The dark gray areas are the original outbreak areas, and then this is where it's spread since 2000. So, very remarkable scientist named Stanley Pressner became devoted to finding the cause of this. Was it a virus? What was it? And painstaking research. He's a physician, but a lot of his background was in protein chemistry. And he managed to isolate in a pure sample some of these abnormal fibers that they saw along with the holes in the brain, the spongiform changes. There's a picture on the right of these fibrils that are under an electron microscope, transmission electron microscope. Very interesting. By 1982, he'd done multiple approaches. He couldn't find any DNA. He couldn't find any RNA in this material. It appeared to be pure protein. And science had never heard of an infectious protein before. They were convinced there must be some nucleic acid in there. And this kept coming up negative. So he originally called it a prion. Then he found out there was some small bird in Europe that was called a prion, so he changed it to prion for proteinaceous infectious protein. I call it prions. I'm giving the lecture, so I'll call it prion. But this appears to be the constant finding in scrapie in sheep, chronic Creutzfeldt-Jacobs disease in humans, Kuru in humans, chronic wasting disease in elk and deer, and then something we're going to talk about just now. The normal protein is highly evolutionarily conserved. If we go back from the amino acid structures, we can tell what the structure of the gene is. And whenever you see a protein that's very carefully conserved among different phyla or families of animals, this usually plays a critical biochemical function. We're not really sure what this is. There's some indications that may play a role in the formation of long-term memory. There's some indications that it may be involved in copper transport in the body. But it's evolutionarily important. It's playing a critical role somewhere. What it turned out through these experiments is that there's a normal version, and it's termed a PRP protein, and there's a misfolded abnormal version. They call that the PRPSC, or scrapie protein. And what happens is that, you know those little crystal things you get when you're a kid? And you add the thing and it slowly makes the crystal and, ooh, wow. You know, this is doing the same thing. There's the normal PRP protein. And when it sees an abnormally folded PRP protein, it induces it, kind of like crystallizes it, to undergo a transitional change and form these beta sheets. And these things tend to stick together to form these very long fibrils. And what you see down here are, under an electron microscope, are these abnormal fibrils, all linked together. So just by the normal protein being exposed to this abnormally folded protein, it induces abnormal folding. You can imagine all the screaming and shouting and impossible and this type of thing that was going on. So it was very, very difficult to prove. Anyway, but painstaking research, this appears to be the way it is. And these plaques that we're seeing in histology, whoops, are just these accumulations of these abnormal PRP proteins. The cell has died, and these have been put into the extracellular space. There's the vacuole that's left after the cells committed programmed cell death. And these fibrils are very, very insoluble. And they stay in the tissue. In fact, they're very resistant to digestion with protease enzymes. In fact, they can survive autoclaving. We had a transmission iatrogenic a few years ago in a hospital where the neurosurgical instruments weren't sterilized in a proper fashion. And the protein will survive autoclaving. So anyway, this leads to progressive neuron cell loss and brain damage. These plaques, again, are just abnormal collections of the PRP protein put into the extracellular space. So what we're seeing under the microscope is actually all one thing. You have these plaques, and these are basically just abnormal collections of these fibrils of the abnormal PRP protein. Further research showed that the normal protein could be associated with a number of genetic mutations. This is the PRP gene here, and the different mutations that have been described in the literature. And their location on the PRP gene. Many different mutations have been identified, and it's usually at positions 102, 117, and 198. And there's a couple different forms of the disease. The first one, GSS, is an inherited dementia. It was reported in 1936. Resembles Creutzfeldt-Jacobs disease, but the cerebellum, the little brain, is more involved. And these were spontaneous mutations. It can occur, or they can be inherited. It takes a number of years to develop this. Intracerebral injection into monkeys. This condition develops within 18 months. So what we have here is phenomenal. It's a strictly genetically inherited disease that's able to be transmitted infectiously. Again, you can imagine all the trauma this caused to classical scientists. So it was finally accepted. A Nobel Prize was issued to Stanley Pressner. And a new class of infectious disease was established. Because of the sponge-like appearance of the brain tissue, the disease was given the term transmissible spongiform encephalopathies, or TSCs. And again, it remained a backwater neurological curiosity, small number of people working on it. And then the gloves came off. In 1985, I was at Oxford at the time. There was an explosive outbreak in the United Kingdom, England. It began with a local veterinarian, Dr. Colin Whitaker, who was attending to a Holstein cow on a farm in Kent in England. The normally placid animal had become quite aggressive, trying to chase you around, uncoordinated, staggering, often falling down, and eventually died. Over the next few months, other farms in England reported the same thing. Entire herds began to uncontrollably stagger and drool in the fields as they tried to graze, eventually dying en masse. The animal autopsy showed that they suffered from the spongiform degeneration in their brains. Disease spread throughout the United Kingdom, and over the next 9 years, thousands of cattle either die or they were intentionally slaughtered and burnt. And there's some of the funeral pyres going on. The problem with this, if you bury them in the ground, they still remain infectious. You have to burn them to charcoal. Panic and collapse of the British beef industry. Almost 1 million cattle, it's estimated, were infected with prions. And it was termed mad cow disease because of the aggressive nature in the animals as part of the disease course. And that seemed to calm down, and then public panic ensued in 1996 when human cases started to appear. These appeared as a variant form of Creutzfeldt-Jacobs disease with a very rapid clinical course. And in contrast to classical Creutzfeldt-Jacobs disease, this started to appear in younger people. All age groups, peak in young adults, and a very, very rapid clinical course until death, and very extremely variable signs and symptoms. Some patients developed incoordination or loss of balance. Some went blind. Some developed severe learning deficits and personality changes before death. This is a diagram of the age incidence. In yellow, as you can see, the younger age of onset of this, and again, a very rapid progression of the disease to death. All patients showed a slightly different pathology from classical Creutzfeldt-Jacobs disease. Fewer plaques, larger rounded halos of spongiform change, and again, this very, very rapid clinical picture and onset to death. This was all different than classical CJD. It turns out, further research shows that you need to have a bit of a genetic predisposition to getting this variant of Creutzfeldt-Jacobs disease. And this is within the normal PRP protein itself. Methionine M is at position 129 in the amino acid chain that makes up the protein, or alternatively, the variant has valine as the amino acid in that position. So this gives us three genetic possibilities: people who have the methionine form, people that have the valine form, and people that are heterozygous, one from mom, one from dad, and they have both types. So what it looks like, if you have a PRP protein, normal PRP protein, with methionine, this makes this protein sensitive to being changed into the abnormal prion form. The variants of CJD, 100% of these cases have been homozygous, that is, you have two copies of each gene, and both of these had methionine at that codon position. Unfortunately, 18 million people in the United Kingdom carry this genetic predisposition. So where's the epidemic? What happened to the epidemic of variant Creutzfeldt-Jacobs disease? It didn't seem to appear. Only 177 overt cases of rapidly developing variant Creutzfeldt-Jacobs disease have been recorded to date. And in 2013, some very clever pathologists and scientists decided to go back and look at archival lymphoid tissue. Everything removed in surgery goes to pathology, and it gets embedded and changed into a tissue slide, and the pathologist reads it. So they went back and they looked at appendices that have been surgically removed. Half of these are still sort of normal. And they looked at tonsils from tonsillectomies and adenoidectomies. And they started to find positive samples, indicating probably these people were exposed to mad cow disease. It's in their bodies, it's in their lymphoid tissue. And from this survey, there's about 27,509 carriers of Creutzfeldt-Jacobs disease. They had looked at 32,441 archived samples. 16 were positive for the prions, with an overall prevalence of 493 per million in the UK population. So there is a subclinical carrier state, and we don't know what's going to happen. The incubation for this thing can be very prolonged. In classical Creutzfeldt-Jacobs disease, it can be up to 40 years. In Kuru, it can be up to 50 years. So we have no idea right now whether these carriers nothing will happen, or they will go on to develop CJD or variant CJD at some time. So the UK could still be decades away from an actual pandemic. Where did this thing come from? Why did it suddenly appear? Well, the rendering industry it gives us a lot of products. Cattle, pigs, everything. When it goes to the rendering plants, everything's melted down, and it's used for a variety of products. You'd be amazed. Everything from lipstick to pharmaceuticals. It's multi-billion-dollar industries. in the world. I've been to these plants. It's quite horrendous. But one of the things that happens is that this residue from the bodies is made into a fortified animal feed for cattle and for pigs. They're also used for supplements. Anyway, to make a long story short, what happens is that you're feeding pigs to pigs and cows to pigs and cows to cows. And in the early days of this, there was a solvent extraction step that seemed to be very efficient at removing any abnormal prions that might have spontaneously arisen in the herds. There was a big explosion in one of these rendering plants, and they got rid of the solvent extraction step, and the stuff made it into the food chain. And they won't bring it back, the solvent extraction. That's over. That's not going to happen. The head of the rendering industry is in Alexandria, Virginia, a little bit south of Washington, D.C., almost a suburb. And they're emphatic that they're not going to bring back solvent extraction. So, what do we know at this time? These prions are part of the transmissible spongiform encephalopathy disease process. We've identified these abnormal prion proteins in fish, sheep, mink, cow, deer, and cats. At the same time mad cow disease was going on, there was an epidemic of mad cat disease from the cat food. Pigs can be infected with the prion from sheep, from scraping. Transmission of prion proteins so far has only been proven to occur between cows and humans. There's 5 known strains of human prions, and these cause 5 recognizably different human diseases, each with its own clinical presentation and its pathology under the microscope. These are Creutzfeldt-Jacobs disease. This can be, you can be born with it, the propensity to develop this, or it can be acquired with time and a random mutation. There's the variant Creutzfeldt-Jacobs disease, VCJD. There's Kuru, familial fatal insomnia. This is inherited, and usually in your 30s, you lose the ability to go to sleep and you rapidly die. And then this GSS syndrome. An infection chain begins with eating contaminated meat, and the prion proteins are taken up by the lymphoid tissue that lines your bowels. From there, it starts making its way to the central nervous system. It amplifying in numbers along the way. And you can see this is a tissue culture slide. The abnormal prion proteins are outlined in red. And we think possibly in a process similar to rabies. You're bitten on the hand, and it takes a number of weeks for the rabies virus to actually travel retrograde up through your nerves to get into your brain. This allows us to quickly give you the vaccine and tell your immune system you need to get rid of this virus. And it's the only example I can think of where the vaccine is actually the treatment. But it's because it takes weeks to get up there. Children that are bitten by a dog on the face and we don't have the dog, we tend to vaccinate them right away because it's a shorter distance to the brain than a periphery. So it may be the same sort of mechanism here. It's traveling through the afferent nerves, from the vagal nerves innervating the bowel all the way back up to the brainstem. The net result is the same. There's a normal appearing sagittal section of the brain, coronal section of the brain on the left. and superimposed next to it is the brain from Creutzfeldt-Jacobs disease. So there's diminution of both white matter and gray matter. And then the normal tissue appearance at the top, and then the abnormal spongiform encephalopathy at the bottom. These plaques disrupt the normal brain architecture, and they induce programmed cell death. There are prion strains. This protein is similar in all mammals, but different species, a change of only one amino acid in this structure can give you a different pattern of disease. So Kuru tends to affect, outlined in blue, the cerebellar area of the brain. Variant CJD tends to affect the thalamus and the cerebellum. GSS tends to affect the brainstem, difficulty in swallowing, fatal familial insomnia, frontal lobe and thalamic areas, and then CJD sort of global. The appearance under the microscope can be a little diagnostic, whether these plaques are florid or not, whether they're occurring at the synapses, the protein deposition, or small plaques, big plaques. They're all sort of linked together. They're variants. And this had confused things for a number of years. So what do we not know about the TSEs? 2019 in Nature Communications, the prions were detected in rodent skin samples as early as 2 weeks after intentional infection. It was transmitted through uninoculated rodents housed alongside the inoculated ones. It was transmitted direct contact or through fomites. And we found this in humans as well. The proteins are in the skin and they can be shed into the environment. We have no idea the clinical significance of that at this time. Anyway, protective measures were established after the 1985 BSE, bovine spongiform encephalitis, BSE outbreak in England, United Kingdom, and the epidemic passed. All cattle had to be tested before entering the food supply. They made it a national notifiable disease in all species, cats, pigs, sheep, whatever. All suspected animals were slaughtered and incinerated without access to the food chain or rendering. There was a ban, total ban on feeding bovine offal to man, animals, and birds, because it ends up as chicken feed as well, some of these pellets. And ban on feeding ruminant protein to ruminant animals. Now, pigs are not considered to be ruminants. This comes to the question of downer cows. A downer cow is livestock that cannot stand on its own, and they're therefore killed and put down. How do we reliably monitor these downer cows? Thousands of them, estimated each year, if they're not presented for observation and testing at meat packing plants. In 2000, the USDA announced that it would exclude downer cattle from the National School Lunch Program. So, like, you can't have a hamburger for lunch at school, but you go home and have one. This doesn't make sense, and it's indicative of the bureaucracy at work again. 27th U.S. Secretary of Agriculture, Ann Veneman, she chose the former chief lobbyist for the National Cattlemen's Beef Association as her chief of staff. The former director of public relations for the Cattlemen Association was her official spokesperson. And one of her Mad Cow Committee appointees had been paid by the beef industry to testify against Oprah Winfrey. She had brought up the question on her program about mad cow disease and beef. A similar conflict occurred in England and everybody got fired in the Department of Agriculture. They were gone the next day, and independent leadership was put in, and a safety agency was created. We didn't do that here. In 2003, the first domestic case of mad cow disease in North America appeared. It was a downer cow. It almost, well, it did make it into the food chain. The USDA vet inspected the case, thought that the cow had a calving injury. The carcass was processed and the meat sent out to 8 states and the territory of Guam before the test came back confirming mad cow disease, and 143 million pounds of beef had to be urgently recalled. The Agricultural Secretary emphatically announced a week later that the USDA will ban all downer cattle from human food chain. The USDA's own Office of Inspector General in 2006 released a report criticizing the USDA for violating its own downer cattle ban. They inspected 12 slaughter plants and found 2 still slaughtering down cattle for human consumption. This is not being taken as seriously as it probably needs to be, especially in light of the carrier status of these 13,000 cases in the United Kingdom. The USDA response to this issue has been a little bit disturbing, to say the least. Japan and England test every slaughtered cow for TSE. The USDA tests only about 40,000 of the 35 million cattle slaughtered in the U.S. annually. And we continue to have sporadic cases here. One case of BSE out of 40,000 tests done each year. It could be due to a natural, spontaneous mutation, a background. But it still leaves 34.85 million cows untested. Overall, the USDA, this has not been its most shining moment. National tracking system is still not in place. There's loopholes in the laws governing animal feed. It's a can't happen here sort of attitude. Thorough testing, they say it's too difficult. And this has been more of a public relations sort of exercise than anything efficient. Currently, inspectors are allowed to determine on a case-by-case basis the disposition of cattle who go down after having passed an initial visual inspection. New York City was fairly proactive in this. In 2001, they instituted an enhanced Creutzfeldt-Jacobs disease surveillance in New York City, and TSEs were added to their reportable disease code. Over a 3-year period, the Department of Health in New York City examined cases, 16 suspected cases in New York City area residents, 6 at autopsy were confirmed with Creutzfeldt-Jacobs disease, and 10 met clinical criteria but no testing done. To diagnose this, you have to have an open brain biopsy and look at the material. At least that was the case. So they came up with a figure of Creutzfeldt-Jacobs disease in their area of 0.75 per 1 million population. And this is what's been the normal for years and years and years. So you would think that if we do have a problem with this, the incidence of Creutzfeldt-Jacobs would be going up. In 2006, a new type of prion strain appeared in U.S. cattle. It's called the L type. It's orally transmissible to monkeys through infected muscle tissue. Classical BSE is not. And the cattle that develop this type of TSE don't have the classical overt symptoms as with mad cow disease. It's called bovine amyloiditic spongiform encephalopathy. I don't know why they don't come up with something a little shorter than all these long names. But these animals appear as downer cows. It's not the classical aggressive nature and staggering. The question is, are prions in the U.S. meat supply, or have they been? The USDA, FDA, and CDC argue that the answer is no, because the incidence of Creutzfeldt-Jacobs disease is not increased, and it's stable at about 1 human case per million population. That is 300 new cases a year. The truth is, we don't know how many cases of Creutzfeldt-Jacobs disease there are in the U.S. It's not a notifiable disease. Is it on the increase in the U.S.? You look at the classical data of 1979 to 2009, it looks like no. But data is misleading, because we now know that a number of misdiagnosed cases of Alzheimer's disease are actually Creutzfeldt-Jacobs disease. Alzheimer's is now the 8th leading cause of death in the U.S. and affects an estimated 4 million Americans. When everyone in this room knows somebody that has Alzheimer's disease, okay? That means it's significant. All of us know somebody. Several studies have shown that about anywhere from 3 to 9% of people that are diagnosed with Alzheimer's actually have Creutzfeldt-Jacobs disease on brain biopsy. CDC continues to maintain that normal incidence is 1 case per million people in the USA of CJD. If we look at the annual new Alzheimer's disease cases diagnosed in 2018, it's roughly estimated at 500,000 new cases. This is going up like this, by the way. It's a very steep slope. If just a minimum of these cases, 3%, are actually CJD on autopsy, that means that the actual annual incidence in 2018 could have been 15,000 cases a year, not 1 case per million, 15,000 cases per million. So look, this is a number of studies have confirmed this misdiagnosis of Alzheimer's as CJD. Is there a mad pig disease? We've been feeding cows to pigs, pigs to pigs, pigs to cows, cows to cows. The PRP protein between pigs and humans has a very, very close homology. Closer than that between cows and humans. Efficient transmission of scrapie, the sheep, to pigs both orally and by injection, show pigs can be infected by both sheep and bovine prions. American farmers typically kill pigs at just 5 months of age before any of these infected animals would have time to show overt symptoms of gait disturbances, which is normally around 55 to 77 weeks. Again, repeated papers on this. Biomedically, it's common sense to say that there has to be a mad pig disease. Hundreds of thousands of downer pigs have arrived at U.S. slaughterhouses, and this cross-cannibalization has been occurring. The prions must have infected or entered the human and cattle food chain multiple times as they make this meat bone meal supplement. USDA counters this by saying old sows show no signs of TSE. Are old downer sows tested before going for rendering? No. There have been a small number of clusters of human CJD linked to pork, reported in the past. The first study found that 1/3 of CJD patients had eaten cow or pig brains with a greater preference for hog brains. Well, this is significant because not everybody has hog brains for breakfast. Okay? My dad used to do this. It's yuck. The second study suggests a dose-response relationship for processed pork. Compared to the people that don't eat ham, bacon, chops, ribs, scrapple, you include these in your diet, you're 10 times more likely to develop CJD. This is ridiculous. We've all been exposed, period. Okay? What about chronic wasting disease? There's been a recent report, and hunters are confirmed that they have been infected with CJD as a result of their hunting and eating elk or deer. CDC says there's no provable link, but it still tells hunters to avoid eating deer and elk brains, spinal cord, eyes, and spleen. Yet, the prions are identified in the muscle tissue of these animals. Oral transmission has occurred between chronic wasting disease. You feed this meat to the chimpanzees, they come down with it. We're getting closer to a sensitive blood assay for Creutzfeldt-Jacobs disease. This work needs to continue. And this represents a new type of emerging infectious disease, a zoonotic protein that's transmissible to man. Where do we find out? How do we get out of this mess? Well, in the United States, I think we need to establish an untouchable medical task force. We didn't take the same steps that the UK did in safeguarding our meat supply. It's been overtly influenced by the cattle, pork, and rendering industry. CDC isn't doing much about it. And I think we should set up a scientific and medical task force, multidisciplinary scientists, well-funded and equipped, free of outside influence. And one of the first things we should do is probably repeat this UK study using archival tonsil and appendixes. If a large-scale study, 40, 50,000 samples, show nothing there, then we probably don't have anything to worry about. We got away with it. But this is something that can be done tomorrow if it was funded. That's about all we want to say on that. But we need to start taking this nationally seriously, especially with these carriers that have been shown in the UK. Okay, I'm tidying up here, but I've been asked a couple questions. This current outbreak of Ebola in the Congo, and we've let a number of Congolese into the United States. And the question was, is this safe or whatever? We recognize a post-Ebola virus syndrome. In other words, you survived the infection, but you have multiple problems, and it goes on for years, and I don't think you're ever going to quite be right. Joint and muscle pain, lethargy, chest pain, hearing loss, hair loss, memory problems, anxiety attacks, vision loss. You can go blind with uveitis. The problem is persistent virus. A 2017 study found the virus in the semen of some men after more than 2 years following recovery from an acute Ebola infection, probably making this the ultimate STD in the world. Its relative Marburg has been sexually transmitted to wives back in the end of the 60s when it was first discovered. Recently written up in the literature, a 39-year-old female nurse from Scotland was working in Sierra Leone. She contracted Ebola. They managed to save her with intensive antiviral therapies. She was discharged eventually with undetectable Ebola virus RNA levels in her peripheral blood. She was readmitted to hospital 9 months later with symptoms of acute meningitis and an acute Ebola virus relapse. The virus was present in the fluid around her brain, the cerebral spinal fluid. She was treated with an experimental drug by Gilead Sciences and again recovered. Researchers from the National Institute of Neurological Disorders and Stroke are doing a 5-year follow-up study of 1,500 Ebola survivors. They'll be evaluated every 6 months, tracking any clinical relapses or the appearance, viral persistence in parts of their body. This lady had infectious virus in her cerebral spinal fluid. There was none in her blood. And after 14 days treatment, that was beaten down. The problem is, this starts to fundamentally redefine what we know about Ebola virus infection. And the paper said that vigilance should be maintained in the thousands of Ebola survivors for cases of relapsed infection. The potential for these cases to initiate new transmission chains is a serious public health concern. And this came out of the National Health System in the UK. That's their highest medical authority. We've recently allowed a number of Congolese refugees into the San Antonio, Seattle, Reno, and other areas. But it's never been really made clear what medical screening had been done on these individuals. I've heard none to all the way to thoroughly screen for everything. But if you have Ebola antibodies, obviously these are people that you want to constantly rescreen in case there is. Anyway, that's the word on that. Are there any questions?
You have talked about the weaknesses that UK has already put into place. I'm very free market, and like limited government, it seems like a good selling point would be to certify all beef or pig as prion-free.
Well, it would be.
Worth pay for it.
It would be. You know, I was over there when all this happened, and they got a grip of this quickly. Every cow is tested. There's new technology coming out that can make this very affordable and very quick. It's not going to disrupt things. And it just doesn't seem to be being done.
Well, I would certainly pay extra
I would
to have
too.
it free, even if it costs more. Yeah.
I would too.
When you say that like a number of cases of Alzheimer's or of CVCJD have been misdiagnosed, do we know anything about the differences between the amyloid plaque of Alzheimer's patients and?
Yeah, it's different amyloid.
Is that why
in
it's hard to-
Alzheimer's, it's the beta amyloid.
Yeah.
But here's this whole thing where we just tripped into a whole new category of diseases.
Yeah.
We're finding synuclein in Parkinson's disease patient. It's a misfolded protein that's occurring in the substantia nigra. There's about 10 of these things now that are, each one is a different protein that's now misfolded and causing disease. So the proteins are different.
Oh, is that what the misdiagnosis is? Is it easy to tell the difference? Like,
Well, we've got good monoclonal antibodies now, and we can stain this brain tissue and actually see, you know, what protein this is.
Yeah.
But it takes a while.
Yeah.
The fact that these prions are in the peripheral blood, because I was at Oxford during this time, I could never give a blood transfusion. Well, look, we've all been exposed, okay?
Yeah.
If there was something serious going on, it would be apparent now. But these things change. Mad cow changed into variant mad cow. So you've got to keep the surveillance up. This is all that I'm saying.
Excellent talk, by the way, and it's very
Thank you.
informative and systematic and methodical. It's very informative.
Thank you. Hi.
Hi, Steve. Thank you. You just used apoptosis programmed cell death, and I'm wondering if there are two kinds of apoptosis, because like in my studies of low-level radiation cells, they have...
Yes,
okay,
there is.
So I like to think of cells being recycled because it's the organism that's alive.
This is what we think, this necroptosis.
Oh, okay. Yeah.
This is a combination, and
There
we
we
think
go.
this is what this low-dose radiation protocols, morning, afternoon, low-dose radiation for terminal cancer patients, and you're seeing remissions.
Oh, yeah.
We think that you're training the natural killer cells with this, and it's a combination of necrosis and apoptosis.
Okay. because
And this
we-
is getting presented in the right form to the antigen-presenting cells that you're training the new generation of natural killer cells. So a very good question.
We have to do something about the 2 million red blood cells we recycle every second of our
Yeah.
lives. So to say that it's, death is, to me, a mental gymnastic. Thank you.
Great talk, Steve, but I was
Thanks.
a little bit confused because when I saw that you're talking about mad cow disease, I was expecting to hear something about the Mad 4 in Washington.
You mentioned that they test every cow. How long does this take, or what kind of a process is it?
I'm not real sure. This is, it takes a little bit of time. The brain samples have to be section processed and immunostained. If you set up an assembly line, you could do it in 3 days. Well, they recall the beef. They've actually, I don't look at the British much anymore.
In the UK, they had the rendering process and
Yeah.
the solvent extraction
Yeah.
failed. Do we have that solvent extraction process here?
We had it here, but they're not doing it anymore, and they're not going to do it. It all stopped worldwide. I've been to these rendering factories. I've spent a lot of time at rendering factories for other reasons. And you're there at night, and you think it's like plastic bags, and you look over, and it's this mound of dead pigs and cows and everything. Like, really, like 10 feet, 15 feet high, waiting the next morning to go get onto the conveyors and go in and be rendered, these carcasses. It's in everything, from lipstick, pharmaceuticals, binding agents, all this. It's very efficient use of animal, but when you put it into the animal feed and feed it back to the animals, you're feeding cows to cows, cows to pigs, pigs to pigs, pigs to cows. And we're kind of worried about, it also goes into chicken feed. We know there's chicken prions. Nobody's even looked at that. So look, we've all been exposed. I don't worry about it. I'm having a hamburger tonight. But what I'm saying is this wasn't handled properly. And we keep dodging bullets, don't we? How many times are we going to be given another chance? This stuff needs to be taken seriously. Yeah, we know how to do this. It's just it doesn't seem to get done. Anyway, thank you very much.